Hello and welcome, dear students. Today we are going to discuss about the topic polymerase chain reaction or PCR. The main objectives of today's lecture are to know the basics of polymerase chain reaction or PCR, to know the history of polymerase chain reaction, to discuss the procedure of PCR, to know the different variants of PCR, to understand the amplification and quantification of DNA, to discuss the medical and diagnostic applications of PCR, and finally to know the forensic applications of PCR. Dear students, let's start with the introduction first. The polymerase chain reaction or PCR is a method widely used to rapidly make millions to billions of copies, that is complete or partial, of a specific DNA sample. PCR technique was invented in 1983 by the American biochemist Kary Mullis. Kary Mullis and Michael Smith developed other essential ways of manipulating DNA, were jointly awarded the Nobel Prize in Chemistry in 1993. PCR is fundamental to many of the procedures used in genetic testing and research, including analysis of ancient samples of DNA and identification of infectious agents. Using PCR, copies of very small amount of DNA sequences are exponentially amplified in a series of cycles of temperature change. The majority of PCR methods rely on thermal cycling. Thermal cycling exposes reactants to repeated cycles of heating and cooling to permit different temperature-dependent reactions, specifically DNA melting and enzyme-driven DNA replication. PCR employs two main reagents: primers, which are short single-strand DNA fragments known as oligonucleotides that are complementary sequences to the target DNA region, and a DNA polymerase. In the first step of PCR, the two strands of the DNA double helix are physically separated at a high temperature in a process called nucleic acid denaturation. In the second step, the temperature is lowered and the primers bind to the complementary sequences of DNA. The two DNA strands then become templates for DNA polymerase to enzymatically assemble a new DNA strand from free nucleotides. As PCR progresses, the DNA generated is itself used as a template for replication, setting in motion a chain reaction in which the original DNA template is exponentially amplified. Almost all PCR applications employ a heat-stable DNA polymerase, such as Taq polymerase, an enzyme originally isolated from the thermophilic bacterium Thermophilus aquaticus. If the polymerase used was heat susceptible, it would denature under the high temperatures of the denaturation step. Before the use of Taq polymerase, DNA polymerase had to be manually added every cycle, which was a tedious and costly process. Applications of the technique include DNA cloning for sequencing, gene cloning and manipulation, gene mutagenesis, construction of DNA-based phylogenies, or functional analysis of genes. Amplification of ancient DNA, analysis of genetic fingerprinting for DNA profiling, for example, in forensic science, and parentage testing and detection of pathogens in nucleic acid tests for the diagnosis of infectious diseases, diagnosis and monitoring of genetic disorders. Dear students, let's know the history of polymerase chain reaction. The heat-resistant enzymes that are a key component in polymerase chain reaction were discovered in the 1960s as a product of a microbial life form that lived in the superheated waters of Yellowstone's mushroom spring. In 1971, Gelclep and co-workers first described a method of using an enzymatic assay to replicate a short DNA template with primers in vitro. However, this early manifestation of the basic PCR principle did not receive much attention at the time, and the invention of the polymerase chain reaction in 1983 is generally credited to Kary Mullis. Mullis summarized the procedure. Beginning with a single molecule of the genetic material DNA, the PCR can generate 100 billion similar molecules in an afternoon. The DNA polymerase is initially employed for in vitro experiments. Presaging PCR were unable to withstand high temperatures. So the early procedures for DNA replication were very inefficient and time consuming, and required large amounts of DNA polymerase and continuous handling throughout the process. The discovery in 1976 of Taq polymerase, a DNA polymerase purified from the thermophilic bacterium Thermus aquaticus, which naturally lives in hot 50 to 80 degrees centigrade environments such as hot springs, paved the way for dramatic improvements of the polymerase chain reaction method. The DNA polymerase isolated from Thermophilus aquaticus is stable at high temperatures, remaining active even after DNA denaturation, thus obviating the need to add new DNA polymerase after each cycle. This allowed an automated thermocycler-based process for DNA amplification. Dear students, let's now understand the principle of PCR. PCR amplifies a specific region of a DNA strand, the DNA target. Most PCR methods amplify DNA fragments of between 0.1 and 10 kilobase pairs in length, although some techniques allow for amplification of fragments up to 40 kilobase pairs. The amount of amplified product is determined by the available substrates in the reaction, which becomes limiting as the reaction progresses. A basic PCR setup requires several components and reagents, including a DNA template that contains the DNA target region to amplify, a DNA polymerase, an enzyme that polymerizes new DNA strands. Heat-resistant Taq polymerase is especially common as it is more likely to remain intact during the high-temperature DNA denaturation process. Two DNA primers that are complementary to the 3'3' ends of each of the sense and antisense strands of the DNA target, that is DNA polymerase, can only bind to and elongate from a double-stranded region of DNA. It also requires deoxynucleoside triphosphates or DNTPs, sometimes called deoxynucleotide triphosphates, or nucleotides containing triphosphate groups, the building blocks from which the DNA polymerase synthesizes a new DNA strand. It also requires a buffer solution providing a suitable chemical environment for optimum activity and stability of the DNA polymerase. It also requires bivalent cations, typically magnesium or manganese ions. Mg2 positive is the most common, but Mn2 positive can be used for PCR-mediated DNA mutagenesis, as a higher Mn2 or manganese 2 positive concentration increases the error rate during DNA synthesis, and monovalent cations, typically potassium ions. The reaction is commonly carried out in a volume of 10 to 200 microliter in small reaction tubes, like for example 0.2 to 0.5 ml volumes in a thermal cycler. The thermal cycler heats and cools the reaction tubes to achieve the temperatures required at each step of the reaction. Thin-walled reaction tubes permit favorable thermal conductivity to allow for rapid thermal equilibrium. Most thermal cyclers have heated lids to prevent condensation at the top of the reaction tube. Dear students, let's discuss the procedure of PCR. Typically, PCR consists of a series of 20 to 40 repeated temperature changes called thermal cycles, with each cycle commonly consisting of 2 or 3 discrete temperature steps. The cycling is often preceded by a single temperature step at a very high temperature, for example, more than 90 degrees, and followed by one hold at the end of the final product extension or brief storage. The temperatures used and the length of time they are applied in each cycle depend on a variety of parameters, including the enzyme used for DNA synthesis, the concentration of bivalent ions and DNTPs in the reaction, and the melting temperature of the primers. The individual steps common to most PCR methods are: first is the initiation. This step is only required for DNA polymerases that require heat activation by hot start PCR. It consists of heating the reaction chamber to a temperature of 94 to 96 degrees centigrade or 98 degrees centigrade if extremely thermostable polymerases are used, which is then held for 1 to 10 minutes. The second step is the denaturation. This step is the first regular cycling event and consists of heating the reaction chamber to 94 to 98 degrees centigrade for 20 to 30 seconds. This causes DNA melting or denaturation of the double-stranded DNA template by breaking the hydrogen bonds between complementary bases, yielding 2 single-stranded DNA molecules. The third step is the annealing process. In the next step, the reaction temperature is lowered to 50 to 65 degrees centigrade for 20 to 40 seconds, allowing annealing of the primers to each of the single-stranded DNA templates. 2 different primers are typically included in the reaction mixture, one for each of the 2 single-stranded complements containing the target region. It is critical to determine a proper temperature for the annealing step because efficiency and specificity are strongly affected by the annealing temperature. A typical annealing temperature is about 3 to 5 degrees centigrade below the demelting temperature of the primers used. Stable hydrogen bonds between complementary bases are formed only when the primer sequence very closely match the template sequence. During this step, the polymerase binds to the primer template hybrid and begins DNA formation. Another step is the extension or elongation step. The temperature at this step depends on the DNA polymerase used. The optimum activity temperature for the thermostable DNA polymerase of Taq polymerase is approximately 75 degree to 80 degrees centigrade. Though a temperature of 72 degree is commonly used with this enzyme. In this step, the DNA polymerase synthesizes a new DNA strain complementary to the DNA template strain by adding free DNTPs from the reaction mixture. The precise time required for elongation depends both on the DNA polymerase used and on the length of the DNA target region to amplify. With each successive cycle, the original template strands plus all newly generated strands become template strand for the next round of elongation, leading to exponential or geometric amplification of the specific DNA target region. The processes of denaturation, annealing, and elongation constitute a single cycle. Multiple cycles are required to amplify the DNA target to millions of copies. The final step is the final elongation. This single step is optional but is performed at a temperature of 70 to 74 degrees centigrade for 5 to 15 minutes after the last PCR cycle to ensure that any remaining single-stranded DNA is fully elongated. This is followed by final hold. The final step cools the reaction chamber to 4 to 15 degrees centigrade for an indefinite time and may be employed for short-term storage of the PCR products. To check whether the PCR successfully generated the anticipated DNA target region, also sometimes referred to as the amplifier or amplicon. Agarose gel electrophoresis may be employed for size separation of the PCR products. The size of the PCR products is determined by comparison with a DNA ladder, a molecular weight marker which contains DNA fragments of known sizes which runs on the gel alongside the PCR products. Dear students, let's talk about the optimization of PCR. In practice, PCR can fail for various reasons such as sensitivity or contamination. Contamination with extraneous DNA can lead to spurious products and is addressed with lab protocols and procedures that separate pre-PCR mixtures from potential DNA contaminants. Specificity can be adjusted by experimental conditions so that no spurious products are generated. Primer design techniques are important in improving PCR product yield and in avoiding the formation of unspecific products. The usage of alternate buffer components or polymerase enzymes can help with amplification of long or otherwise problematic regions of DNA. Both the running parameters, for example temperature and duration of cycles, and the addition of reagents such as formamide may increase the specificity and yield of PCR. Dear students, let's discuss about the variants of PCR. First, the quantitative PCR or qPCR. It is used to measure the quantity of a target sequence commonly in real time. It quantitatively measures starting amounts of DNA, cDNA or RNA. Quantitative PCR is commonly used to determine whether a DNA sequence is present in a sample and the number of its copies in the sample. Quantitative PCR has a very high degree of precision. Quantitative PCR methods use fluorescent dyes such as cyber green, Eva green or fluorophore containing DNA probes such as TACMAN to measure the amount of amplified product in real time. It's also sometimes abbreviated to RT-PCR or real-time PCR. The second one is reverse complement PCR or RC-PCR. It allows the addition of functional domains or sequences of choice to be appended independently to either end of the generated amplicon in a single closed tube reaction. This method generates target-specific primers within the reaction by the interaction of universal primers, which contains the desired sequences or domains to be appended and RC probes. Reverse transcription PCR or RT-PCR, it is used for amplifying DNA from RNA. Reverse transcriptase reverse transcribes RNA into cDNA, which is then amplified by PCR. RT-PCR is widely used in expression profiling to determine the expression of a gene or to identify the sequence of an RNA transcript, including transcription start and termination sites. If the genomic DNA sequences of a gene is known, RT-PCR can be used to map the location of exons and introns in the gene. The 5' end of a gene corresponding to the transcription start site is typically identified by race PCR, that is rapid amplification of cDNA ends. Dear students, let's discuss the applications of PCR. PCR has numerous applications, for example, selective DNA isolation. The PCR allows isolation of DNA fragments from genomic DNA by selective amplification of a specific region of DNA. This use of PCR augments many ways, such as generating hybridization probes for southern or northern hybridization and DNA cloning, which require larger amounts of DNA representing a specific DNA region. PCR supplies these techniques with high amounts of pure DNA enabling analysis of DNA samples even from very small amounts of starting material. Other applications of PCR include DNA sequencing to determine unknown PCR amplified sequences in which one of the amplification primers may be used in Sanger sequencing, isolation of a DNA sequence to expedite recombinant DNA technologies involving the insertion of a DNA sequence into a plasmid, phage, or cosmid depending on size or the genetic material of another organism. PCR fingerprint methods have high discriminative power and can be used to identify genetic relationships between individuals such as parent-child or between siblings and are used in paternity testing or disputes. This technique may also be used to determine evolutionary relationships among the organisms. Dear students, let's discuss about the amplification and quantification of DNA. Because PCR amplifies the regions of DNA that it targets, PCR can be used to analyze extremely small amounts of sample. This is often critical for forensic analysis when only a trace amount of DNA is available as evidence. PCR may also be used in the analysis of ancient DNA that is tens of thousands of years old. Quantitative PCR or real-time PCR or qPCR methods allow the estimation of the amount of a given sequence present in a sample, a technique often applied to quantitatively determine levels of gene expression. Quantitative PCR is an established tool for DNA quantification that measures the accumulation of DNA product after each round of PCR amplification. There are two methods for simultaneous detection and quantification. The first method consists of using fluorescent dyes that are retained non-specifically in between the double strands. The second method involves probes that code for specific sequences and are fluorescently labeled. An interesting technique combination is real-time PCR and reverse transcription. This sophisticated technique called RT-qPCR allows for the quantification of a small quantity of RNA. Through this combined technique, mRNA is converted to cDNA, which is further quantified using qPCR. This technique lowers the possibility of error at the end point of PCR, increasing chances for detection of genes associated with genetic diseases such as cancer. Dear students, let's now understand the medical and diagnostic applications of PCR. Prospective parents can be tested for being genetically carriers, or their children might be tested for actually being affected by a disease. DNA samples for prenatal testing can be obtained by amniocentesis, chronic villus sampling, or even by the analysis of rare fetal cells circulating in the mother's bloodstream. PCR analysis is also essential to pre-implantation genetic diagnosis, where individual cells of a developing embryo are tested for mutations. Many forms of cancers involve alterations to oncogenes. By using PCR-based tests to study these mutations, therapy regimens can sometimes be individually customized to a patient. PCR permits early diagnosis of malignant diseases such as leukemia and lymphomas, which is currently the highest developed in cancer research and is already being used routinely. PCR is very useful in the medical field since it allows for the isolation and amplification of tumor suppressors. Dear students, let's discuss the infectious disease applications of PCR. PCR allows for rapid and highly specific diagnosis of infectious diseases, including those caused by bacteria or viruses. Characterization and detection of infectious disease organisms have been revolutionized by PCR in many ways. The other one is the human immunodeficiency virus, or HIV, is a difficult target to find and eradicate. PCR tests have been developed that can detect as little as one viral genome among the DNA of over 50,000 host cells. So infections can be detected earlier, donated blood can be screened directly for the virus, and the effects of antiviral treatments can be quantified. Some disease organisms, such as that for tuberculosis, are difficult to sample from patients and slow to be grown in the laboratory. So PCR-based tests have allowed detection of small numbers of disease organisms, both live or dead, in convenient samples. Viral DNA can be detected by PCR. The high sensitivity of PCR permits virus detection soon after infection and even before the onset of disease. Such early detection may give physicians a significant lead time in treatment. Diseases such as pertussis or whooping cough are caused by the bacteria Bordetella pertussis. This bacteria is marked by a serious acute respiratory infection that affects various animals and humans and has led to the deaths of many young children. PCR is an important testing tool that can detect sequences within the gene for the pertussis toxin. Recently, we had seen the example of COVID-19 when this PCR was used as a confirmatory test for diagnosis. Dear students, let's talk about the forensic applications of PCR. The development of PCR-based genetic fingerprinting protocols has seen widespread application in forensics. In its most discriminating form, genetic fingerprinting can uniquely discriminate any one person from the entire population of the world. Minute samples of DNA can be isolated from a crime scene and compared to that from suspects or from a DNA database of earlier evidence or convicts. Forensic DNA typing has been an effective way of identifying or exonerating criminal suspects due to analysis of evidence discovered at a crime scene. PCR is a very powerful and significant analytical tool to use for forensic DNA typing. For example, a single human hair with attached hair follicle has enough DNA to conduct the analysis. Less discriminating forms of DNA fingerprinting can help in DNA paternity testing. DNA from unidentified human remains can be tested and compared with that from possible parents, siblings, or children. Dear students, let's know about the research applications of PCR. PCR has been applied to many areas of research in molecular genetics. PCR allows rapid production of short pieces of DNA even when not more than the sequence of the 2 primers is known. The task of DNA sequencing can also be assisted by PCR. Known segments of DNA can easily be produced from a patient with a genetic disease mutation. PCR has numerous applications to the more traditional process of DNA cloning. It can extract segments for insertion into a vector from a larger genome which may be only available in small quantities. Sequence tag sites is a process where PCR is used as an indicator that a particular segment of a genome is present in a particular clone. An application of PCR is the phylogenetic analysis of DNA from ancient sources such as that found in the recovered bones of Neanderthals or from frozen tissues of mammoths. A common application of PCR is the study of patterns of gene expression. Tissues or even individual cells can be analyzed at different stages to see which genes have become active or which have been switched off. Dear students, let's now conclude the lecture. It is fairly simple to understand and to use PCR and produce results rapidly. The technique is highly sensitive with the potential to produce millions to billions of copies of a specific product for sequencing, cloning, and analysis. qRT-PCR shares the same advantage as the PCR with an added advantage of quantification of the synthesized product. Therefore, it has its uses to analyze alterations of a gene expression levels in tumors, microbes, or other disease states. PCR is a very powerful and practical research tool. The sequencing of unknown etiologies of many diseases are being figured out by the PCR. The technique can help identify the sequence of previously unknown viruses related to those already known and thus give us a better understanding of the disease itself. If the procedure can be further simplified and sensitive non-radiometric detection systems can be developed, the PCR will assume a prominent place in the clinical laboratory for years to come. Dear students, it was all about today's lecture regarding polymerase chain reaction or PCR. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.